Hello everyone, it's a pleasure to speak to you about my research topic on this providing more robust and sensitive peptide identification results using the protein feedback mechanism in Comet. I am Chen Zhou from the Hong Kong University of Science and Technology. As we all know that in the bottom-up proteomics, peptide identification is crucial to the success. So this is the overall pipeline of this experiment. We first have our entry samples, we digest them into the peptides, and then we send all the peptides into the LC-MS to generate the MS1 spectrum and the MS2 spectrum. And during these experiments, we typically need to use the software to interpret all the MS2 spectrum. So the Comet is a classic one that provides these reasonable results, but its output quantity is sensitive to the input parameters such as this MS1 tolerance, MS2 tolerance, and database size. Turning these parameters back and forth to obtain the optimum is of great importance, but really time-consuming. So we first did an experiment. We used a synthetic dataset, and then we changed the MS1 tolerance from 0.02 to 1 Dalton, and MS2 tolerance from 0.2 to 2 Dalton. And then we tried different combination of this MS1 and MS2 to run the same dataset. We further plotted the PSM number of these different combinations and see that this result shows that their output PSM is very sensitive to this different combination of this tolerance. So you see that this is the value plot of this result. You can see that in the suitable parameters, we can have 550 results. But if you are unlucky to have a bad parameters, you only have less than half of the results. And also, we fixed our MS1 and MS2, and then we only changed the database size here in this plot. So we add more and more irrelevant protein sequence in our database, and then we try to run the Comet again and again. So you can see that the performance or the PSM numbers also change to this database change. So you see we also have this violin plot here. You see in the worst situation, we only have 350 results, and in the best situation, we can have nearly 650 results. So this is the problem when we run the data analysis in the Comet. So to address this problem, we proposed this protein feedback mechanism. I'm going to use this here to demonstrate the basic concept behind this. First of all, we have a query spectrum, MS2 spectrum. We use the X correlation to run this from find out their candidates. We then sort all of the candidates by their scores in a descending order. The first one, the second one, and so on and so forth. So we first retrieve the similar score one, for example, this FKAVSAK versus this NYQEAK. They have a similar score here. And then we retrieve their corresponding proteins. We can see that for this FKAVSAK here, it only has one peptide identified in the whole data set here. But we go to another peptide NIQEAK here. Its corresponding protein, he has a lot of sibling peptides identified in the whole data set. So that we can see that if we jump out of the current spectrum and using the global information about their sibling peptides, we can see that actually NYQEAK is more trustworthy, so that we need to add some additional information into the current spectrum and then adjust their score. And maybe we should report this NYQEAK as the correct PSM instead of the highest scored ones, FKAVSAK here. So this is the basic mechanism behind the protein feedback. And then we implement this idea into the Comet and generate this Comet PFM. So you can see here, this is the overall pipeline of it. I'm not going to the details for the sake of time. If you want to see everything, you can go to our parser here. You can see that there is a loop here. This is basically why we call this method feedback. So we designed a Comet PFM here, and we want to see the performance between the Comet PFM versus the Comet. So we did the same experiment again. First, we changed the MS1 and MS2 tolerance and use different combinations to run the Comet and Comet PFM. So because these data are the synthetic data set, we can actually have their ground truth. So we calculate their FDP, the false discovery proportion. You can see that in this figure here, the Comet PFM, it doesn't have much difference to the Comet. But in the bottom figure here, you can see that the PSM numbers here, the Comet PFM always have a better PSM number. But more importantly, the Comet the surface of Comet PFM is very flat. But in contrast, this comet, the surface of this comet is irregular, shape in an irregular shape, and also we can have this violin plot to see that comet PFM always have a more concentrated result, and also a better sensitive, yes. But in contrast, the comet here is more dispersed and less sensitive results. So the same experiment applies to the database as a variable. So you can also see that when we change the database, this FDP doesn't change much, but also we have the Comet PFM always performing better than the Comet, and it's less sensitive and it's more robust to the parameter changing here. And also you can see the volume plot here. The PSM number of the Comet PFM is still more concentrated. So in the end, we did some experiments on the real data side, and we observed that the Comet PFM can systematically identify more PSMs, and also on average, we can identify 37.6 more results than the Comet. So that's all for the poster. Thanks for listening, and you're welcome to contact me at this email, and welcome to our poster.